and this is Dr. Timothy Davies from the University of Durham. So Tim completed his PhD at Cambridge University, where he studied the biochemistry of kinesin involved in cell division. He moved to Warwick with his lab for a short period at the end of his PhD, before starting a postdoc in New York at Columbia University. During his time there, he explored cell division using a classic model system, so C. elegans zygotes. And in the second part of his postdoc, he focused on investigating if cells in the same organism divide in different ways, and if it's these projects that he's going to discuss with us today. And lastly, Tim is currently setting up his lab at Durham University to continue his work, and he's going to talk to us a little bit about career paths at the end of his talk. So over to you.
Thanks. Yeah, thanks, Rebecca. First of all, thanks so much for the opportunity to speak. It's really kind of exciting. So from that introduction, I think you can tell that I've obviously been away from the UK for quite a long time. And I'm just now came back in January to set up my own lab. So it's quite nice, even if it's only in a virtual sense, to give talks at other universities and kind of begin to establish, connections with people here in the UK, which, to be honest, this is not a good postdoc move. I neglected a lot when I moved away. So anyway, having said that, I'm going to talk to you today about my work looking at cytokinesis in the C. elegans embryo. So I think you could see from the movie that was playing, the cell division is this dynamic and rapid process. I think, let's see if this plays. Here we are. Now, I think cytokinesis is important. Hopefully it's kind of the importance of it is fairly obvious. Now it's critical for all life, basically. The fact that all cells come from other cells was one of the earliest kind of discoveries in cell biology, when people started building microscopes that allowed them to observe cells directly. Furthermore, cytokinesis is required for development and tissue homeostasis. So obviously cell division is important to generate, a multicellular organism, but in many animals, that's going to continue throughout life to, keep tissue homeostasis intact as cells die and are damaged. And finally, cytokinesis failure can be pathogenic in a variety of different ways. I've listed a few here. Cancer is often the primary one that people think about when they're doing cytokinesis and mitosis research. That is because if you have one cell that divides properly, you end up with these two mononuclear daughters, which is great, and development or homeostasis can continue. However, if you have cell division failure, you end up with this one cell containing two nuclei, and importantly, double the number of centrosomes often. And this can become highly oncogenic. And in the case of these mice, cells were caused to fail in a dish just once, and then they were injected and could be oncogenic in a range of different circumstances. So that's kind of how we classically think about cytokinesis intersecting with disease, but I'm particularly interested in the idea that there might be cell type specific variation in cell division. So that kind of idea came from fairly recent findings that some cell types are specific to mutation of cytokinesis genes. So on the left here, we have these multinucleated erythroblasts. These are cells that have failed to divide earlier in the blood lineage, basically. And this is caused by a patient that has a mutation in an important cytokinesis gene. Now, this is a very, very mild disease because only this specific cell lineage fails to divide. Despite the fact that all of the other cells in the patient have that same mutation, it seems to only be having an effect on these cell type. Now that's surprising because the gene is essential for cell division in, we thought, all cells. So, there seems to be this idea that maybe some cells are more sensitive to hypermorphic or weak mutations because the protein reactivity is highly required. And then on the right here, we can see examples of what I'm terming natural cytokinesis failure, where we have these binuclear hepatocytes where the two nuclei in a single cell are indicated by these arrows. And then now, unlike the kind of oncogenic potential I discussed on the previous slide, in some cases, this occurs due to cytokinesis failure as part of aging or tissue differentiation, basically. There's kind of controversy about the cause, and it likely is different between different cell types. But the idea is that, cells fail to divide, and that gives rise to these binucleate cells. And that can be a good thing or a bad thing, and it can just be part of normal aging. But to be honest, the more you look into it, the larger the number of cell types there are that contain these binucleation events, kind of much wider than I had expected when I started to look into it. So my hypothesis that I've kind of built up during my postdoc, and then I'm bringing into my, running my lab, is that the textbook view of cell division is a little bit incomplete, and there's more complexity than we typically admit. So often, if you see examples of cell division in textbooks and even in papers, they'll be drawn on the left, where you have a single cell dividing in a symmetrical manner. It doesn't really have any reference to its cell fate. And it's often dividing out of any context, any three-dimensional or even often two-dimensional context. Whereas we know that's not true in a multicellular organism where we have cells with different fates, different structures. Some cells are touching other cells, as you can see here on the right. Some are kind of like highly different in size and shape. So my kind of hypothesis is that, and the question I want to ask is, how does cytokinesis vary with cell identity? So, when I'm talking about cell identity, I'm meaning things like cell type, cell fate, the environment it's in and the contact it's making with a matrix or with other cells, and so on. However, in this case, I'm interested in cells that have the same genetic background, but have these different identities. So having kind of maybe said it's overly simplistic, the kind of textbook model of cytokinesis is still useful to understand what's going on. So we have here a cell that's in the process of dividing. It's just gone into anaphase. The chromosomes are going to be equally inherited by each cell. This is being done in conjunction with the mitotic spindle, the microtubule structure that acts to both segregate the chromosomes and also acts to signal to the outside of the cell. Again, this is a little bit of a simplistic version because there were also inhibitory signals going on. But the idea is that the mitotic spindle positions the formation of a contractile ring. This is made of actin and myosin and goes around the cell equator. This is a kind of a biomechanical structure that will be able to deform the cell and cause it to divide. And if we just zoom in on this for a second, we can see, again, this is highly simplistic, but these structures are going to be composed of three proteins I'm going to discuss. These actin filaments that are being assembled by formin, a protein that sits on the end of the filaments and pulls in monomeric subunits, causing this to extend. And then also there's myosin here that cross-links and acts to constrict this structure. And now when the myosin is active, it can constrict this and cause the structure to shrink. Because this is cross-linked by many other proteins that I'm not discussing here, both to itself and to the membrane and to the cell cortex, we get this deformation in shape and the cell will begin to divide. So we can image all of these things, which is quite nice. So formin and myosin on the left and right are shown using a tag, a genetically encoded tag at the endogenous locus where GFP was added using CRISPR to the gene. So we can see the contractile ring, especially in this bottom panel. This is kind of the end-on view where the contractile ring can be seen kind of in progress, beginning to constrict the cell. And then in the middle, we have actin. Now, actin doesn't like being labeled directly by GFP. So rather, we have an actin binding probe that's fluorescently tagged and localizes to these actin filament structures. So then we can kind of take a step back. And again, this is highly simplistic, but we can say, OK, there's a schematic here where formin is important for assembling these linear actin filaments. And then myosin acts to constrict them. And together, they drive cytokinesis. So the question I'm going to be asking later on in my talk really is, how does cytokinesis vary with cell identity? And can we see any differences in these pathways depending on the cell we're looking at? But first of all, I want to introduce my model. So I don't know if this is going to play. Oh dear, some of these movies are not playing. I'm sorry about that. Anyway, okay, so this was going to be a worm crawling along. So I'm sure many of you have seen them. This is the C. elegans worm. I appreciate that at Bath you have people working on other worms, so I need to clarify. This is the kind of more boring one that a lot of people use. But I think it's great. It's a simple multicellular system. So we have cells with different types, which is obviously going to be important if we're interested in looking at how a cell's identity intersects with the behavior that we're interested in. We have genetic tools that, this is a great thing about using a tool that a model that other people have been working on for decades. There are lots of tools available to us. They're optically clear, so we can do nice imaging. And then when we come on to the embryo in particular, we have this stereotyped morphology, so we know the cells, we know the identity of the different cells, and we can also image them going through multiple rounds of division just on a fluorescent microscope. And finally, there are conserved cytokinesis genes that with fast-acting temperature-sensitive mutants. Now, what that means, I'll come on to in a minute, but it's really been crucial for kind of all the work I'm talking about today. So to begin with, I'm going to talk about the molecular requirements for cell division in the one-cell embryo. Then I'm going to look at how they might change in a multicellular context. And then for this talk, I'll just add in a little bit at the end about applying for postdoc positions to give you a few minutes of my thoughts on how to go about that. Okay, so we can jump to the one-cell zygote. Oh man, I'm really sorry these videos are not playing very well. Okay, this was going to be an example of a nicely synchronously dividing embryos where they all divide at the same kind of speed and rate. So I'm going to just skip that and say, okay, we can now, a bit of a description of these temperature sensitive mutants I talked about earlier. So often the genes that are involved with cell division are highly important. And if you cause a complete knockout, often the organism will never develop because they're required for division events and development. So instead we use these conditional temperature sensitive mutants. So these are often point mutations in the gene of interest, such that at the permissive temperature, the protein is functional and you get a wild type like behavior. So if the mutation is in a cytokinesis gene, at the permissive temperature, the cell will divide correctly. However, at the restrictive temperature, the protein now gives a loss of function phenotype. And if it's important for cell division, the cell will fail to divide and we'll end up with these binucleate cells I talked about earlier. Now, importantly for our work, these are very rapid. So they're not kind of temperature dependent decay because of some protein unfolding response, but rather, protein inactivation occurs in about 20 seconds and reactivation occurs on the same time scale when you drop the temperature of the sample back down to the permissive. So we developed a tool where we could combine kind of microscope imaging and these mutants, where a device called the Therminator passes either cold or warm liquid kind of over the embryo, changing the temperature, turning it either off or on, depending on whether the protein is hot or cold. So then kind of for the beginning part of my postdoc, I was interested in seeing whether, oh, some noise there. I'm interested in seeing when these proteins were important for division. So we did that by just looking at this time series and saying, can we inactivate the protein at blocks of time to see what is the outcome on cell division? And we have mutants in both formin and myosin, as well as a whole load of other genes that we can test to see when are these proteins required. Today, for simplicity's sake and time, I'm just going to show you the results from formin. And I hope this movie plays. No, it doesn't. That is very frustrating. Okay, I might just exit PowerPoint and then maybe relaunch it and see if that makes a difference. How can I do this here? Sorry about this. This has not.
You could show us the slide not sort of full, like size and see if it plays.
Let me try that. Can you see it now?
I can see it, yeah. Let's see if it works.
No, why is it not working? Okay, that's fine. I can just describe it and we can move on. That's okay. Sorry about this. This is a learning curve for me because, as I say, I've not used Teams before. So basically what we did was we inactivate the protein at different times during division. So this little schematic along the top here, if anaphase onset, when the chromosomes begin to separate, is indicated by this little arrow. We could inactivate it shortly after anaphase onset, a bit further after anaphase onset, or much later. And the phenotypes you would have seen is that this one fails to even form a contractile ring, this one forms a contractile ring but fails to divide, and this one divides successfully. So therefore we can say that at these two time points, the early and mid time points, the formin activity was required, whereas later on it wasn't required. And then doing this multiple times and with multiple different genes, we could build this kind of landscape or this timeline of when protein functions are required. Now, this was kind of cool because all these proteins were known to be essential for cell division and had been known by other studies, but now we could actually work out when these things were functioning. And this is important because people build models of the way that the proteins interact with each other. And yet sometimes they don't know if their models are correct because, they're requiring them to be interacting, throughout the process, whereas we could show that, for example, the formin is only required early on, presumably because it needs to build the contractile ring. And then once it's built, there's enough actin there that the myosin can keep constricting it and divide the cell, whereas the motor protein myosin is required throughout division. So having said all that, I can now move on to how these requirements might change in a multicellular context. And I'll explain, why the jump happened and how that first half of the project set me up really well for the second. Okay, so we can come back to this schematic and see, okay, let's just focus now on formin and myosin and then say, if we look at this very simplistic model, maybe we can have cell-specific factors playing some role that will, either enhance or prevent the requirement for formin and myosin for cell division to occur correctly. So the focus of my study for the second half of my postdoc was in the 4-cell stage embryo. So this is a kind of a simple model. It's really nice because it really all we have to do is wait for 20 minutes from the movies I was showing you earlier, where you have the 1-cell division occurring. But now we have a multicellular system. Each cell has its own identity. If we just go on here, we can see that, the 4 different cells at this time point come from this lineage map. They're going to give rise to different tissues in the adult. For example, the EMS cell gives rise to the intestine. The P2 gives rise to the germline. Muscle cells come from all of them, basically. But they can be identified by their lineage. So we can watch this live and know that the AB cells derive from the anterior blastomere, for example. But also, just by looking at the embryo, we can identify them based on their position. So I would just say it again. They're the ABA, ABP, EMS, and P2 cells. I appreciate that you're probably not going to need to know these again in the future. So I've tried to keep them color-coded so it's obvious which is which throughout the rest of my talk. So now we can come back. And as I said, the first half of my postdoc really set me up nicely for this, because now instead of inactivating these proteins using these mutants, during cell division, we can actually do it during development. So we can allow the proteins to be functional by keeping them at the permissive temperature for these first 2 rounds of cell division. And then once we have developed into this multicellular system where you have 4 different cells, we can inactivate the protein by shifting the temperature and observe, does cytokinesis occur properly? This would be much more difficult if we were using a tool like RNAi, where the effect is much longer lasting and would likely disrupt some of these earlier divisions as well. So here we have, I'm just going to focus on this EMS cell. If it divides successfully, we're going to get these 2 mononucleate daughters. And if it fails to divide, we're going to see this binucleate cell at the bottom. Just see if I can make my laser pointer a bit better. There we are. These 2, this binucleate cell at the bottom. So it's very simple to score, did division occur correctly or not? So now we can go back and say, okay, are formin and myosin required for division to occur properly? So the data I'm going to be showing for the rest of this talk is going to consist of some of these bars where we have cells, a bar representing each individual cell. So here we have ABA, ABP, EMS, and P2. The little cartoon below it is going to show you the context. So these are all in the intact embryos. Now the bar represents the percentage of cells that either complete or fail to divide in green or gray, and then they're going to be clustered based on the genotype. So here we have control cells, they all divide, which is great. That would be a worry if they didn't. And now we can do the same thing using these myosin temperature sensitive mutant embryos and observe that all of them now fail to divide. So again, this is great. It suggests that we're actually inactivating the protein based on this temperature method, and myosin is required for all the 4 cells. So that wasn't particularly surprising, given that myosin has been ascribed to have an important role in cell division in basically every model that people have looked at. So then when we did the same experiment using the formin mutant embryos, we saw something quite surprising. So first of all, 2 of the cells failed to divide 100% of the time. Now, this was what we expected, basically, given the data that I tried to show you in the 1-cell stage. This protein is important for division in the model that we previously used. However, in these 2 cells on the right, the EMS and the P2, they were able to divide about 30 to 40% of the time, even after we inactivated this protein. So this was a shock because, 2 of the cells are dividing. or rather, some cells are able to divide, and also it's specific to the identity. This isn't just, all 4 cells are able to divide, but 2 of them are specific. So it gives rise to this question, why does formin inactivation only affect some cells? So to investigate this, we thought maybe the simplest solution is just there's lots of formin in those 2 cell types. Maybe if it's overexpressed 4 or 5 times and we're not inactivating it fully, there's enough residual activity left to allow those 2 cell types to divide. So when we looked at this, we tagged this protein using CRISPR. So hopefully we're getting normal kind of expression levels and look to see the localization. And in each case, the protein localized to the contractile ring as we were expecting, as you can see in all these micrographs. But the level of intensity seemed to be approximately the same when we did a line scan across the cell. They all form a peak of about the same intensity at the cell equator. So we don't think that's an effect. At least there's no strong difference in the level of formin between these cell types. So we can move on and think of another option. So then we thought maybe, okay, cell-specific factors might just be assembling linear F-actin, and then we're bypassing the requirement for this formin because something else is making the actin filaments. So we did kind of quite a bit of work to rule out this possibility. While I was doing this work, a few years before we published it, another lab did a really nice single cell transcriptomic study where they isolated individual cells from the early embryo and did single cell RNA-seq and looked to see, what transcripts are present and are they localizing to different cells. And there are many transcripts that localize to specific cell types. However, none of them seem to be important for assembling actin filaments. And especially when we look for actin assembly factors that might be enriched in both the EMS and P2, we couldn't find any. So then we thought, well, there are other formins. I've been focusing on this one formin, Cyc1 is the name, throughout my talk, but there are 5 or 6 others. Now, they haven't been ascribed any role in cell division, but we thought, okay, maybe they're having some compensatory function. But when I knocked them down by RNAi in this temperature-sensitive mutant background, cell division wasn't disrupted. So we thought, okay, that seems to be unlikely, but let's just have a look directly at the actin to see what's going on. This is, we're a microscopy lab predominantly, so, take a look if you can't understand what's happening. So we can do this, as I mentioned earlier, using these probes that bind to actin filaments. So we can see in the control at the top here, in the 1 cell stage zygote, we have this really nice localization to the equator, just as the cell is about to divide. I should comment that these puncta that you see dotted around the cells are due to ARP23, and they're not the linear filaments that are going to be productive for cell division. So when we look in the mutant embryo, we see, okay, this equatorial enrichment has completely gone. Now, this is great because, again, these cells are all going to fail to divide. So in this 1 cell embryo, we have this correlation between, equatorial localization of actin and cell division. And when that equatorial localization is gone, the cells will fail to divide. And then we looked at the 4 cell stage, and I'm just showing data here for the EMS and P2 cells because they're the ones we're really interested in to work out what's going on. And we can see that in controls, it's a little bit more tricky, but where these brown arrows are, you can see there's some enrichment at the cell equator. So we do think, okay, there's a contractile ring and we're able to measure it just about. But then when we look in the mutants, that's gone basically. So now there's no equatorial enrichment. It's a little bit spotty here and we see some localization of these puncta, but we can't see what we would consider a normal contractile ring. And again, we did just a line scan across the cell to try and quantify this. We see that there's a nice peak in the EMS and P2 control cells, and this is lost in the formin mutant in blue at the bottom here. So this was a surprise because, some percentage of these cells that don't show any equatorial localization for actin are going to divide. So actin probes can be a little bit temperamental and they can label subsets of actin populations. So we use 2 other ones as well. Plastin and LifeAct both label actin filaments as well. And in no case could we see equatorial localization of actin, in this mutant, even though a significant number of these cells are going to divide. Okay, so now we can go and say, well, we don't think that this kind of branch of the pathway seems to be important, but that's too big a jump for us. The idea that linear actin isn't important for cell division seemed, to be honest, kind of ridiculous. So we wanted to back it up using a different method. Now, up until now, I've been using these temperature-sensitive mutants. Now, these aren't that common in other systems, so we thought let's try an approach that lots of other people will be familiar with, and that is using latrunculin A. Now, LAT-A inhibits incorporation of free actin monomers into the end of the fiber. So we think it acts in a fairly similar way to disrupting forming function. We wanted to see, okay, what effect does this have on cell division? So traditionally, people haven't used drugs that much with cell division studies in C. elegans because there's an eggshell that surrounds it that makes it, impermeable to drugs, and drugs can't, access the cell. Fortunately, about 10 years ago, some really nice papers came out where people now are able to permeabilize this by knocking down proteins that help to assemble the eggshell, basically. And a well-named PERM1 RNAi is able to permeabilize this. So then you can permeabilize the embryo, you can transfer it to a growth media that contains the drug of choice, in our case, latronculin, and also a dye. And the dye is important because you can monitor whether the embryo was permeabilized or not by the presence of the dye inside the embryo. And then, you score the embryos that were permeabilized to see, did adding this drug affect cell division? So again, we can come back to the data in a similar manner to I showed you previously. So we see that, we can see that on the left here, these controls always divided successfully. So we don't think we're messing anything up by this permeabilization method, at least not in the first few rounds of cell division. And then when we look at low doses, I have to admit, but when we add latronculin in a range of different concentrations, we see that the EMS and P2 cells are able to divide more frequently than the ABA and ABP cells. And this is true over a range of different latronculin concentrations, basically. Now, if you add too much, you abolish cell division in all cases, presumably because you're just completely disrupting the actin cytoskeleton. But this does suggest two things. One is that, or it raises two questions, I guess. Firstly, how do these cells divide without a normal contractile ring? And secondly, why is this effect specific to certain cell types? And I should just say, which of these questions you're more interested in probably depends on if you're a cytokinesis person, where you might be more interested in question 1. And if you're a developmental biologist, you might be more interested in question 2. Now, of course, hopefully they will both link up and have the same answer at the end. We're working on that, and I'll show you some of the progress we're trying to make to get there. Okay, so then up until now, I've been talking about maybe there are cell-specific factors, but what can we call these more specifically? So we can break them down into extrinsic or intrinsic. So intrinsic are things like polarity and inherited factors based on asymmetric cell division. So for example, germ granules are inherited by the P2 cell that's going to give rise to the germline, these RNA-rich granules. So the intrinsic factors are things that will be present even when you don't have any adjacent cells. Whereas extrinsic factors are things like cell-cell contact mediated by cadherin, and various signaling pathways where the cells signal to each other. Another nice thing about working on C. elegans is that all of these factors have been, or not all, I should be careful about saying that, but a lot of these factors have been identified and these pathways have been dissected over previous decades of genetic work. So we thought we'd take a bit of a sledgehammer approach and just get rid of all of the extrinsic factors. So we can do that using fairly simple, although quite fiddly technique, where we remove the eggshell, and then at the 2-cell stage, we just physically separate the cell into 2. So we have the AB and P1 cells. We let these divide again, and then again separate them into the ABA, ABP, EMS, and P2 cells. So now we have all 4 cells separate. I should comment at this point that the difference between ABA and ABP are thought to be entirely due to extrinsic factors, which we've now removed, and we can't tell them apart. So I'm going to call them the AB daughters, and we can just pull their results, basically. Okay, so here we have all 4 cells. Now I hope this movie plays. Okay, great. So on the left, we have the AB daughters. In the middle, we have the EMS, and on the right, we have the P2 cell. So these cells are dividing asynchronously. The EMS and P2 divide later, with the P2 last. Also, you'll note that the P2 is dividing asymmetrically. Now, these are all features that we know should be true based on work done in the embryo, and they're dependent on these intrinsic factors. So it looks like the intrinsic factors are still present. So we can do the same thing now where we isolate cells from a formin mutant embryo. And then when we observe cell division, we can see now that some of the cells are failing to divide, which is what we expect. I'll just note that the P2 cell here that divides properly, it still has this difference in cell cycle timing between its progeny. So again, these intrinsic features seem to be correct. okay, this is just one embryo, but when we do this lots of times, we see something quite interesting. And that is that the EMS cell now is not able to divide when it is isolated. And this is in contrast to when we looked in the intact embryo when it was dividing 30 to 40% of the time. So this suggests that maybe some extrinsic factors are present. And when we look at the P2 cell, basically division occurs at the same frequency, whether it's isolated or intact in the embryo. So we can come back to our schematic and say, well, it's got a bit more complicated than we thought, to be honest, but there seem to be extrinsic factors that are important for division in EMS and intrinsic factors that seem to be important in the P2 cell. So briefly, the last few minutes of this section of the talk, I'm just going to look at some of these extrinsic factors that might be important. So again, based on decades of previous work, we have these really nice pathways mapped out. We know that P2, the neighboring cell, signals to EMS via Wnt and frizzled, as well as via this receptor tyrosine kinase that signals via SARC, the proto-oncogene. And that's important for various features like cell fate and orientation of the spindle and so on. So we thought, okay, well, let's just keep this signal sending cell adjacent to the one we're interested in to see what happens. So on the left here, we have the AB daughter cells dividing and, they fail. That's not surprising. But here we have an EMS now paired up with its sister cell. And we can see in this example, it gives the game away a little bit, but we now see that the EMS cell is able to divide. So when we quantify this, the controls are all dividing fine, which is great. And then when we look in the mutant embryos, we see that an EMS cell in a pair with a P2 is now able to divide again. The P2 cell divides, at the same frequency, independently of whether it's been isolated or in a pair or whatever. Yeah, so there we are. And then when I did a kind of a technically more complex experiment, but now I isolated the EMS cell, but then paired it up with an AB daughter. So this doesn't send any signal. We didn't see this ability for the cell to divide restored. So that's great. It looks like the P2 is sending some signal, and that's allowing the EMS cell to divide in this mutant background. But you'll notice that the number of cells dividing is a bit lower than we were expecting. The frequency is about 15 to 20%, so half what we thought. And I thought, well, why is this happening? So we think that the signal is being sent from P2. So maybe we can hypothesize that the contact area between the cell is important for this signal to be sent. So maybe the larger the surface area, the larger the signal, or the more likelihood of a productive signal. So when I just measured the area of contact, we saw something quite nice. And that was, if we look in this middle one here, we've separated out the formin mutant cells into the ones that failed to divide and the ones that divided successfully. And we see that the ones that divided successfully had a larger surface area. So again, it fits with this model. It's not kind of conclusive proof, but it fits with the model that this signal is, able to be sent because there's this larger surface area between the two cells. I should just comment that, one big difference we saw actually was the surface area between control and mutant cells. And we think that possibly by disrupting formin function, we're disrupting the actin homeostasis that we know is important for generating things like cell tension and adhesion between the two cells. So, in this mutant background, we've already disrupted formin activity and we're getting rid of some of this ability for it to signal. But in the cases where the signal is being sent, where there is a good chance for the signal being sent, these are the cells that are dividing. Okay, so we thought we'd do another experiment, which is now knocking down some of these signaling pathway components, but in the intact embryo. So now I tried to do various things like Wnt and some of that pathway, and the RNAi didn't work very well. That's something obviously I'm planning on getting going now in my own lab. But SARC is known to mediate some of this signaling. So when we knock down SARC compared with control RNAi treatment, we see this really nice result, which is that the EMS cell specifically doesn't seem to be able to divide anymore. While the P2 cell, is dividing as frequently as it did in the intact embryo previously. So we can come back to our schematic and say, okay, there seem to be cell extrinsic factors that are important for division. These seem to be coming from the P2 cell, and they seem to be being mediated via SARC signaling. To be honest, this doesn't really answer the questions I came into, and this is where, hopefully I'm in a good position to set up my own lab to kind of dissect some of these. But it, does push it a little bit back further up that pathway. So briefly, as Rebecca said at the beginning, I'm setting up my own lab now in Durham. I'm hoping to investigate some of these questions using C. elegans, but then also an in vitro tissue culture system. So initially, I'm planning on using the same 4-cell model I use at the moment. But I'm also really excited about moving into non-embryonic C. elegans tissues. There are these beautiful seam cells that run down the length of the worm, which we can see here. This is just a complete control, just because I think it looks pretty. But we can see these cells dividing. They run in two layers. So we've got the top in purple and the bottom in green, kind of after this has been computationally straightened, dividing. These are a stem cell-like model because one cell divides to give rise to a cell that will divide again and another one that will differentiate. Being C. elegans, this only happens a few times before all the division stops. But I think it should be a cool model because things like Wnt and cell morphology are important for regulating this. So that's kind of one of the directions I'm going in. I'm also really interested to see, can we apply some of the knowledge we're getting in the C. elegans model into a mammalian in vitro system? Maybe you're looking at 2D models or 3D models where, can we mimic a 3D model in vitro and some of the signaling pathways that we see in the C. elegans embryo, and observed as to have the same impact on cell division. For example, disrupting actin polymerization in vitro and then getting rid of some of the pathways we think might be important. Do we also see an effect on the ability for some cells to divide and not others? So anyway, that's all I wanted to pitch. Again, leaving you with these 3 questions. How do cells divide without a normal contractile ring? Why is this effect specific to certain cells? And then now we can add in a third one, which is, not only when we're thinking about forming, but does cell identity affect the requirement for other essential proteins? A lot of the lists of proteins we think of as being essential for cell division are because they've been shown to be important in a relatively small number of models. But as we expand out to different cell identities, we might see a variation in what really is an essential cytokinesis protein. So with that, I don't know, I'm happy to answer any questions. I've got this bit about maybe applying for postdocs that I can jump to. If that would be better, and then take questions at the end, which I don't know if you have any preference on that, but yeah, maybe I'll jump straight to the postdoc stuff and we can take all questions and stuff at the end.
Yeah, that's a good idea. We'll do that. Okay,
so this just take a few minutes, but I really have a passion for helping people apply for postdocs that they think are helpful as they move through their career path. My attitude wasn't very thought out, to be honest, as I thought about applying for postdocs and kind of thinking about how I wanted to direct my scientific career. So I'm trying to, pay it forward a little bit or pay it back or whatever, and maybe give some thoughts and encourage you to begin to think about what you might want to do once your PhD is done, basically. So I think the first important question is, why do a postdoc? I don't think it's helpful and particularly healthy if it's just the default option when you finish your PhD, which I know it is in some cases. So really start to think, maybe you want to learn specific skills. Maybe you want to work in a research environment that you've enjoyed during your PhD. Hopefully you've enjoyed during your PhD. In my case, I was a real fan of microscopy. So the idea of working with specific equipment for my postdoc, fancy microscopes or whatever, was a real motivating factor. Often you'll have a topic of interest that hopefully it might relate to your PhD work, it might not, but in some way you've kind of come up with an idea of something you're interested in. And you want to kind of take that further. Or finally, it might just be that you really enjoy, planning out a problem, a scientific question, working out a way to answer it, and then, enjoying that kind of iterative, kind of independent research, and you want to take it to another level. All of these are great reasons. You might have a million others, that's fine. But I just want to suggest you begin thinking about this, especially depending on the 2 main intended destinations. A lot of people who start a postdoc are planning on going into either industry or academia. I should note there are a whole host of other exciting things you can do. And often people who do a postdoc and then realize maybe that industry or academia aren't for them will move into, this is just a very small list of potential options on the right here. But I think it's fairly true based on my friends and, contact experience that at the start of the postdoc, typically industry or academia are the direction people are aiming for. And if that's the case, it's good to think about it in advance so that you can begin to tailor your postdoc to fulfill some of the requirements you might need to make progress in these 2 destinations, basically. So, hopefully now I'm speaking to some of you who might be early on in your PhD or a bit further on, some of you even who are postdocs, who are thinking about second postdocs or going forward from here. I think, it's important to do some advanced prep. What are your goals? Do you want to have a job in an industrial lab that, you can progress with? Do you want to teach? All that sort of thing. Begin to think about what your goals are for your career. And again, the questions I put earlier, why do you actually want to do a postdoc, and do they align? If you want to work in industry, maybe doing a postdoc so you can learn a specific skill is important. If you want to go into academia, it's so that you can develop your own independent project, so on. But just try to make sure that these things are all aligned before you make a jump, rather than realizing that they're not afterwards. You want to think about the topic of research. Often people move from one model to another, but looking at the same biological process, or they really love working with their model, but they take it to a different biological process. All of these things are appropriate, but it's often useful to have at least a reason for why you're thinking about going in a certain direction. And finally, location and what type of institution might be important. So I did my PhD and undergrad in the UK. So when it came to postdoc, I thought, right, for various personal reasons, I thought I'm done with the UK, I'm moving. So I only applied to postdocs abroad. So again, that starts to narrow down the type of places you might go to. And then also the institution is important. If you're interested in teaching, maybe going to a research intensive, non-university affiliated lab in a research institution might not be so important or a good idea. But if there are these staff scientist roles that are kind of a super postdoc type thing, where you're either attached to a lab or you maybe run a core facility. Doing a postdoc in an institution that has those people might be really helpful for moving into that area too. So as you look for positions and you apply, I think a few things to keep in mind. Again, with your end goals in mind, will this help you? I touched on this before, but if you really want to become a microscope facility manager, which to be honest sounds like an amazing job, you're going to need to get really a lot of microscopy experience during your postdoc. If you want to go into, if you're coming from a worm lab like me, but you want to go into industry, maybe doing your postdoc in a lab that gives you experience with some of the skills that you will need in industry is more important than, fly picking. Worm picking is lovely, but not that many companies require it. And so you might want to move into a slightly different field. Again, make these decisions early on rather than when you realize it's too late. I think, with conversations with friends and colleagues, the number of times people realize they're thinking about something a year later than they should have done is really common. Then you want to think about things like lab size, and just because a lab has 10 Nature papers in the last 20 years, that might look really good. But if there are 30 postdocs, the chances that you're going to be one of those postdocs who gets a Nature paper is pretty low. So you want to be a bit more nuanced in the way that you kind of maybe look at a lab. Also, you might want to be in a big place, but then you're probably not going to get that much interaction with the PI and all that sort of thing. Some of this stuff, I'm sure, but again, as you begin to think about this, I think it's good to bear in mind. Finally, I think in the UK, often a lot of positions are just advertised. So someone has funding for 2 years or 3 years for a postdoc. So, you want to look, and they advertise for that position. So you want to look at length of contract and the specifics of the project. What actual techniques will I be using? Is it very prescribed in the direction, or will I have some freedom to move around? Maybe will it be renewed? All that sort of stuff is important for kind of positions that are advertised for a specific project. And the other option, which is I think maybe a little bit more common in other countries, is cold emailing. So this is what I did. I just emailed 15 different labs that I really liked and asked if I could work with them. And I would guess probably half of them didn't reply, and a couple of them replied and said no, and another few said, oh yeah, okay, come for an interview, and that was enough. So that's a way of doing it. So if you're thinking about that, maybe identify labs you want to work in based on, again, the region you want. If you have a geographical constraint, look for papers that you've read recently that came from the town you want to live in or the university you want to go to, and see if those labs might be appropriate. You can ask around, for friends in your department or faculty to say, do they have suggestions of good labs that will be good at mentoring you and helping you in your career? And then finally, a lot of this comes down to writing a good cover letter. I heard people talk about this all the time, but then as soon as my email got put on the Durham website, I started getting emails too from people who it was clearly copy pasted and they'd sent the same thing to 50 different labs. So if you are going to cold email someone, really make sure you write a proper cover letter or cover email explaining why you want to work in that lab that's clearly personalized to them, and not just copy pasted with 50 other people. It might take a bit longer, but you're at least going to be setting off that relationship on a good foot. Even if they don't have space, you might make a new contact or collaborator. Again, some places will really want to see what you've actually done in the past. So if you don't have a first author publication, which to be honest, in the UK, because of the short nature of our PhDs, can be a bit of a problem, try to see if your boss will let you send them a manuscript or a preprint or that's been posted to bioRxiv or something. Not necessarily you have to do that in your first contact, but think about having that sort of thing available to prove that you've actually done some work in the past. Then funding can be important. So again, if it's a fixed lab, a fixed position, they should have that funding available. Sometimes the lab themselves will have funding kind of ready to go because they have a grant that they've just got. In that case, they might want you to start quite quickly because time is money really, and if they have the money, they want to spend it. In other cases, they might ask you to write for fellowships, which then and often will take a lot longer. So, it can be awkward because if you write out to a load of labs just as you're finishing up your PhD and they say, yes, we'll support you write a fellowship application, it can leave this intermediate time without funding and with nowhere to go. As you interview, I actually asked around a few other friends who've recently got faculty positions about what they would suggest. And this was one of the things that came up, is, check out that the person you're going to work with, who I'm calling in this context your mentor, is going to be good for your needs. Is their management style what you want? These are all things you should be comfortable asking, either to the person directly or their lab once you're kind of interviewing. Do the mentor's goals align with you is really important. I went to a very new lab. I was the first postdoc. And part of that was because I had not very many publications for my PhD. I had one first co-authored paper. So I joined a new lab because I knew that she was going to need to get papers out quickly to get tenure in the US. Whereas a good friend of mine went to a much bigger lab and really struggled because there was no pressure on that lab that he went to publish because they'd been around for years. So it ended up with him struggling for 5 or 6 years before his work was deemed to be of good quality and to be able to be published. So, if you want to, you need to start thinking about that sort of thing as well. And then again, maybe if you're going into industry, it might be important. Look for someone who maybe has already done industrial collaborations and will be able to have an insight into that world. A lot of academics, the only thing they know about, this is maybe an unfair generalization, but often the only thing they know about is how to get an academic job, which can be really important. But if that's not what you're interested in, try and find someone who can mentor you in other areas too. And finally, things like lab culture, are they friendly? Does everyone work 9 to 5 and then go home? Do they work 9 to midnight and then tell everyone how hard they work? Because if you're not that kind of person, you might not particularly enjoy that lab. And then funding and access to equipment, again, are you going to enjoy being in this place? Are you going to have the resources to do well? And then finally, and this is going to be really important for the people who want to go on and run their own research group and maybe become a professor themselves, is, can I suggest new ideas and can I take them with me when I leave? And again, these hopefully are things that your new potential postdoc supervisor will be willing to discuss with you, even at the early stages of having an interview. And one of the guys I interviewed with, he very candidly said, look, while I'm paying your salary, you need to work on the project I'm interested in. But if you get a fellowship, to work on that's a project that's on a slight tangent from what the lab works on now, you can work on it in my lab and then you can take that all with you when you leave. So again, they're the kind of things that maybe you want to be thinking about, because even at the early stages of your postdoc, you want to know that you're aligned and going in a good direction. So briefly, things to do now, I would just really encourage you, think through if you want to do a postdoc and why, because I really enjoyed mine, but as a default option, because you can't think of anything else to do, it might not be that great. Find people to discuss the process with. A peer mentoring amongst students in your department is really powerful. Faculty and postdocs can give you good advice. Visiting speakers even might be willing to respond to emails if you have questions that you think they can help with. So explore areas you might be interested in. Give yourself permission to read outside your PhD. This is tough because I know everyone's super stressed and busy, but maybe even if it's just once a week or once every couple of weeks, skim read a paper that is something you're interested in, but doesn't really have anything to do with your PhD, because it might open up another area of science that you hadn't considered previously. And finally, I think research low information areas. So types of jobs that maybe you haven't heard about before, or what industry are looking for, or specific industrial jobs that you're interested in might be looking for, as well as, how different funding situations work in different countries and so on. I didn't really know anything about the US system before I went there. And actually, there were a few fellowships I wanted to apply for. And to be honest, I didn't hear about them until I had had my postdoc position for too long. So I think, learning as much as you can in advance can also be helpful. And finally, there are lots of different resources. I just want to encourage, I think I've really found Twitter useful. If you're worried about mixing personal and work, make a work Twitter account. That's what I've done, and follow other scientists in your field, especially ones that, some of them are very good at retweeting job adverts and things like that. So that can be a great way of finding out personality a little bit, but then opportunities. And then something that has been really powerful in my career is these new kind of online Slack communities. So there's one called Future PI Slack, which was for a load of postdocs, that a postdoc set up that was discussing how to go about applying for faculty positions, that I found really helpful as I went through that process. But I think now someone has set up a grad student Slack, and this is their Twitter handle. It probably is quite US centric, but maybe one of you could even go ahead and try and create a UK one if there isn't one already, or maybe a UK channel within this to provide feedback about departments and labs and just bounce ideas back and forth. Not all of these ideas will be suitable for everyone, but I recommend giving them a try. They could be quite useful. So then finally, having said all of that, sorry, I feel like I've been talking for hours and hours. Hope it's been helpful. I just want to acknowledge that all of this work was done in my postdoc lab. This is Julie here in the middle. The first half of the talk was done in conjunction with Sean, who was a PhD student. That was all that 1-cell data that the videos didn't play. And then the 4-cell data was done with 2 undergrads, Natalia and Benjamin, who did help me with some of the data analysis and some of those experiments too. So I'd like to finish there. Please, if anyone has any questions, feel free to get in touch. You can ask them now. This is my email if you want to reach out. Maybe you're interested in cell division, or you have a question where you think worms could be useful and you want to collaborate, let me know. Maybe you have questions about cell division as well. Feel free to ask them now, or drop me an email later. So with that, I'll finish and take questions.
Great. Thank you so much, Tim. I'll open the room to questions. And maybe if we start with questions about the talk, and then we can have a few afterwards about career paths. If people want to ask any questions.
If you don't, that's fine. I appreciate I talked very quickly then. Sorry about that. I was aware that I had to put in a few more slides than I thought. So.
I have a small one we could start with. It was about, I'm actually really interested in the inactivation with temperature and using that. So it's not actually something I've seen much of before. Is it quite limiting? Like, is it quite limited in terms of the number of genes that we're able to do that with proteins?
Yeah, really good question. Really good question. So it's been used in yeast for a long time. Okay. First of all, you need to use a species that can grow at different temperatures, right? HeLa cells are not going to enjoy growing with a 10-degree temperature difference. But this works because worms can be propagated at 16 and they can be propagated at 26 degrees Celsius. So you can look to see if there's a difference between them. Now, the way that they're identified is by random mutagenesis. So these are done by mutagenesis screens like you would do to identify mutants kind of in classical genetics. But that does limit us, so we can't design them yet. And we're not quite sure what is happening, to be honest. I'm really interested in collaborating with some maybe kind of protein biophysicists, because what we think is happening is the mutation is disrupting some, hydrogen bond between 2 different amino acids. And then that makes it slightly weaker, so that when you add in the increased thermal energy and you have thermal vibrations, it just causes the protein to unfold slightly and not be functional anymore. And then when you remove that thermal energy, it refolds again. So that's the model, but we don't know. So it is a bit limiting. It's very powerful for the ones that we have access to, but it is a bit limiting in that they're not able to be designed intentionally yet. That would be cool.
Definitely.
Yeah, especially like in some cases there are conserved residues where you have like a kinase that has a temperature sensitive mutant, and then it has another kinase that has, and the residue that's mutated is conserved in the second member of the family. So the idea could be that you can port the mutation from one gene to another and create an artificial temperature sensitive gene. But I've heard reports of people doing that, but nothing published, and I haven't tried it myself. It'd be really cool. Yeah. No time yet, but these things should be easy now because CRISPR should allow us to make those kind of mutations by design in a way that was much easier than it would have been a few years ago. So yeah.
Cool, great. Got any other questions? Feel free to post them in the chat if you don't want to put your hand up and speak. Okay.
That's okay. If they want, don't worry about it. That's fine.
Yeah. Okay. Well,
I
mean, I think you've also got your contact details. You could always everyone should follow on Twitter. I agree with you on the postdoc front. A lot of postdocs are well advertised on Twitter, and I think it's a really good community for looking at progress and things. Oh, Louise has a question.
Highly recommend it. Well, you've got to be careful. There was a big elegans, like a weird elegans like controversy that got kicked off the other day because the editor for a journal made a joke about how they weren't very useless, and it got a bit of a mess. But on the whole, I feel science Twitter at least is a fairly positive place.
Yeah. So Louise, do you want to say yours out loud, or do you want to type it in the box? Louise asked, what was the biggest cultural difference you found between the UK and the US?
Oh, interesting. Interesting. Money. There's more money, I would say. In the I joined a lab that was quite new, which meant that they had a big startup budget. So she could basically employ me for the first couple of years without having to get a grant to do so. So that was actually a real positive about going to a new lab. Often, especially in the US, they might have funding for a postdoc available. So that was quite a shock. And actually coming back here now, I have to remind myself that not that budgets are smaller necessarily, but there maybe is, I was in a kind of a weirdly unique situation as well in the US, probably, where there was more money floating around a bit in a way that probably isn't that typical for the US either. I think the UK, because it's smaller, sometimes that actually is helpful in the sense that it's a bit more collaborative. You know, it's much easier to go and visit someone at a different university when it's just an hour drive than if it's, you know, an hour flight. So that's something actually that I didn't appreciate so much there. But again, it could be because, you know, all of these things are so heavily dependent on your specific lab environment that it's a little bit difficult to make generalizations. But yeah, but I really loved it. And again, I mean, I was in a hugely privileged position that I could travel without having to worry about like caring responsibilities or family and things like that. But as an opportunity to go and experience a different culture, you know, doing a postdoc abroad for a few years was really fun. Highly recommended. Highly recommended.
Cool. And Claire asked, for postdoc applications, are many people successful by cold emailing?
I think so. I would say just be really careful about it. So in this new PI, so I said this Slack group, I'm now in one called New PI Slack, which is for people who have been like a PI, have been a professor for less than like 5 years. And in that, sometimes they have a discussion about it where I think it's becoming, because more, hopefully, good professors that you want to work for are interested in being kind of an equitable place. So now they're becoming better at putting instructions on how to cold email on their website. It's not something I've done yet, but you know, saying, hey, if you're going to apply for a postdoc in my lab, you're welcome, but can you include these documents? You know, stuff like that. And those people are probably quite receptive. But yeah, I think a cold email where you clearly outline why you're a good fit for the lab and make it obvious that you've read some papers by that person and have thought about it, I think have a much higher chance than, you know, kind of randomly spamming 100 faculty heads. But yeah, that's how I got my job. I just cold emailed. And I should say that sometimes it helps if you don't hear back, maybe wait a week and then email again. And if you don't hear then, you know, maybe it's not worth trying. But yeah, I would say try once, then a week later, try again. And see what happens, and just be polite and respectful. And you know, the worst that can happen is they say no, or that, you know, often they don't reply. Some, as I say, about half of mine didn't reply, but most people at least reply to say no. So I recommend it. And to be honest, I think that's something that is a skill that I'm still not good at and I don't enjoy. But at some point, you know, you kind of have to get over that and try and push through. So yeah.
That's great. I don't think we have any more questions unless anyone wants to jump in. But yeah, so if everyone can give Tim a follow on Twitter, and then we'll say goodbye for now.
I didn't do this just to boost my follower. I have
no,
also, you'll be sorely disappointed, but I will try in the run after this to retweet postdoc adverts more than I normally would. So.
Yeah, no, it's great. But yeah, thank you for talking, and
Oh, you're welcome.
hopefully we can all sort of keep collaborating.
Yeah. Oh, well, good luck everyone on finishing up PhDs, and at the moment it's very stressful, I can imagine, for all of you. So yeah, good luck getting back into the lab and trying to get stuff back going again.
Yeah. Cool. Thank you. Bye everyone.